Everybody. So I'm going to present in a few minutes the activity, the technology, and business model of Promethera Biosciences. So can we go to the next slide? Doesn't work? It doesn't work? Yeah? Okay. So let's start by the medical needs we are addressing with Promethera Biosciences. So you see here an example, an illustration of the disease we are targeting today in our clinical trials. You see on the left a premature newborn that has a jaundice due to immaturity of the liver, and it is under UV. It is under UV, in fact, to destroy a very toxic pigment called bilirubin that stick to the skin and provide this yellowish color. This pigment is very toxic for the brain, and to avoid the brain toxicity is under UV. The UV destroyed the bilirubin. Fortunately, after a few days or few weeks, the liver that was immature become mature, and you can relapse the treatment. Unfortunately, in some cases, the issue is not related to the immaturity of the liver. It's a genetic mutation. It's what you see on the right. This little girl has the enzyme that conjugate the bilirubin mutated and has not the ability to excrete this very toxic pigment. In this case, the little girl needs to spend 10 to 12 hours per day all her life under UV to destroy the bilirubin. This disease is called Crigler-Najjar syndrome. It's a very rare disease, fortunately, but there is more than 300 different genetic defects that can affect the hepatocyte, the main cells from the liver. And within Promethera, we want to target these indications. So what we are doing with Promethera is infusing healthy cells coming from healthy donors, because today the only way to cure this treatment outside the UV, the little girl needs to spend 10 to 12 hours per day, is liver transplantations. So we infuse cells, liver stem cells, coming from the liver of cadaveric donor instead of full liver replacements, knowing that for this disease, you don't need the full mass of enzymatic activity to have a normal life. 3 to 5% in general is enough to pass from a very severe phenotype to a mild phenotype. So we call the product HepaStem. So we are currently in clinical trials in phase 1-2 of clinical trial in Europe and Israel. We have closed the enrollment period this month with 20 patients on 2 indications. Urea cycle disease, it's one of the inborn error of metabolism. In this case, when the kids eat proteins, there is accumulation of ammonium in the blood, with, once again, a very high brain toxicity. And the second indication is Crigler-Najjar, the little girl I presented you earlier. So we have orphan drug status for these 2 indications in Europe and in US. So the process, how it works. The raw materials is liver from cadaveric donor in the same system that for liver transplantations. We have a tissue bank in Belgium. We are a Belgium-based company that can collect liver from cadaveric donor that are not transplanted but fit the transplantation criteria. And based on an easy process of collagenase digestions of the liver and centrifugations, we collect what we call hepatocyte bag, which is 95% of hepatocyte and over cells that are the raw materials that we are using in our process to purify and quantify and amplify in very large quantity for the scale-up process the stem cell present in very small quantity in the human hepatocytes. At the end of the manufacturing campaign, we cryopreserve the cells in liquid nitrogen, and when the patient is ready and the catheter, a radiocatheter is in place, we infuse the cells through a syringe and a pulse syringe in the portal vein of the patients having the genetic disease. The healthy cells will repopulate the liver and produce the missing enzyme that is not present in the liver of the patient. So what we do is enzyme replacement, like Johnzyme, Shire, etc., but using the cells as a vector instead of recombinant proteins. We have as well developed some new innovations on the process. We have developed this GMP van. In fact, we can formulate to the last step of thawing and formulations inside this van that has been GMP approved. And we have been able to develop this process. We had to develop this process because we have more than 12 centers in Europe, and we could not have GMP facilities for the towing and formulation in all the centers. So we have developed this mobile formulation unit where the technicians drive the van with the frozen cells in dry shipper inside the van and go to the parking of the hospitals, formulate with a closed system called CPAX the cells, and produce freshly formulated cells to the patients when the catheter is in place in the portal vein. What is important to know as well is thanks to this scale-up process, with one liver we can treat more than 400 patients today, instead of one liver, one patient with liver transplantation. So we really resolve the issue of shortage of liver, which is one of the key issues for this disease. At the top of that, one healthy cells coming from one healthy donor can treat, in theory, any inborn error of metabolism because he has all the enzyme, the activity of the enzyme of a normal hepatocyte. So in terms of pipeline, as I said earlier, we're in phase 1-2 in pediatric indications, in urea cycle disease and Crigler-Najjar syndrome. We are thinking about hemophilia A as a new indications to do some proof of concept in adult this time, maybe next year. And we have in earlier phase in proof of concept in animal liver fibrosis, but here with a very different mechanism of actions where we will produce stem cell to replenish the stem cell pool that has been consumed by the toxic related to alcohol or viruses and to inhibit the fibrosis mechanisms. We have done 2 rounds of financing so far. First round of 9 million euros, including 4 million of grants from the French-speaking part of Belgium. And the second round of financing was done last year of 23.6 million euros with arrival of very interesting new investors like Mitsui Global Investment, the Japanese fund. It's one of the biggest Japanese VC funds. The Shire Strategic Investment Group, the VC funds of Shire, Boehringer Ingelheim Venture Funds, but as well ATMI Life Science. It is a company developing bioreactor for adhering cells, and we're working with them. In fact, we have even transferred the full process using their bioreactor for the scale-up of the manufacturing process, and other smaller players. So we have a team today of 46 people, all people based in Belgium. Main part of the activity of the company, as we have internalized completely the manufacturing, is related to QC, manufacturing and development of methods. 5 people in clinical development. It's even if we have few number of patients, we need a lot of energy for all of these different patients, regulatory affairs and IP, of course. The cells that we are working on, the progenitor of hepatocyte, has been patented, and the patent has been granted in Europe, US, Japan, and Australia, and we're waiting for the other country, but we should come very soon. The team as well is one of the assets, we believe, of the company. We get, of course, the scientific founder, Professor Etienne Sokal, who is one of the key opinion leaders in this area, in liver disease transplantation, is a pediatrician. But we have as well several people coming from GSK vaccine, the vaccine part of GSK. The headquarters is maybe 10 minutes by car from our facilities, and it provides very talented people that knows how to develop and manufacturing biologics. Competitive advantage, I try to summarize in one slide what is the key competitive advantage of product. So it's adult human mesenchymal stem cell committed to the hepatic lineage. So we try to differentiate the cells in fat, bone, or whatever. It never works. The cell just collapses. It only does hepatocytes. It's really engaged already in hepatic lineage. We take the cells from the liver and we reinfuse in the liver, so the risk of having something else is extremely low. We have seen effective engraftment and differentiations in different animal models, but even in human through biopsy of the liver after infusions of the cells. It's an allogeneic medicinal product with low immunogenicity that is related to the mesenchymal origin of the cells that has been proven in vitro study. It's a resistance to cryopreservation, so we can store the cells for years. We have seen in vitro proliferation, in vitro and in vivo with safe phenotype, and we believe as well a longer duration of efficacy due to the capacity of the cells to proliferate in the niche of the stem cells. So we are definitely in the ultra-orphan business model, the model of John's Shire, a very low number of patients and high price. So you see it's very high unmet needs. It's life-threatening disease, pediatric. The only way to cure these patients today is liver transplantations. So for the first one, Crigler-Najjar is super rare disease. We are talking about few hundred patients. We selected this disease because the clinical endpoint is extremely simple. It's level of unconjugated bilirubin in the blood. So you can follow, if there is a decrease, you see an effect. If you don't, if you stay in the same level of hours under the UV. The second indication selected is urea cycle defect. So we have more patients, 3,000, 3,000 to 4,000 in US and Europe. But you see there is a pool of over indication with a higher number of patients that we will develop by extension of indication after the proof of concept on the two first indications. So the cells can be considered as a platform, and the idea is to expand step by step the different indications. So in terms of pricing, I just show a benchmark of the average yearly price for this kind of therapy, $250, and the cost of a liver transplantation in US is just 525,000 euros. So you see there is space for price related to the cost of goods of this kind of therapy. Clinical development plan. So we have spent quite a significant amount of time to develop what we believe are the right protocol for this kind of therapy. It's pediatric, orphan drugs with cell therapy. So what we did, it's two different studies in parallel. One observational study where we follow the patients with the standard treatment of care to follow the natural history and data disease progressions. And in parallel, we do the investigational study where the cells are infused to generate the data. The idea is to strengthen the validity of the result, to see an evolution of the disease and what is the effect of the cells, to strengthen the impact potential of the cells. In this kind of disease, you cannot do placebo. It's completely impossible. And having these evolutions of the disease progressions is the only way to compare with no treatment at all. What we do, it's 3 doses. For cell therapy in general, one of the key questions is which dose. The animal proof of concept are good to do the proof of concept, but for the dose range with xenotransplantation, it's not a good predictor. So we decided to do a dose range right from the beginning with 3 doses. A high dose, mid dose, and low dose, and on 3 cohorts of patients according to the age, the infant, the teenagers, and between these 2 cohorts. Why? Because the liver, the capacity of growth of the liver is different. And maybe the quantity of the cells required according to the age and this growth potential could be different as well. And we do the 2 indications in parallel as we are regrouping the safety. The primary endpoint is safety. Secondary endpoint is preliminary efficacy, so we can regroup the safety of both indications in one platform. So I didn't show the slide because I believe I didn't have time, but the objective is to do second clinical trials in 2015 in US and Europe to pivotal clinical phase 2/3 with 2 doses than in 1 dose to get market approval and to submit for the dossier for both indications 3 years after the initiation of the clinical trials, that could be mid-2015. And we are preparing now the next step of financing that should happen end of 2014 with current investor, of course, but as well with new investors. And we'll be very happy to have US investors because the US part of the development of the company will be extremely important. And I try to summarize here the key elements of the key selling point of Promethera. One element maybe just to underline to finalize is the competitive intensity. We have seen that in this liver disease, for genetic disease, outside us and maybe a German company, there's nobody really involved. So the level of competitive intensity in this area is extremely low. I would like to thank you for your attentions.